Hello, everyone, and welcome to our Clinical Pharmacology Training Network webinar for this week. So I appreciate everyone's joining us. Again, there's CME offered. The information will be provided at the end of the webinar for you to obtain that credit for attending this session. It's my pleasure today to introduce Dr. Jun Yang from St. Jude's Children's Research Hospital. Dr. Yang is a professor and endowed chair in pharmacogenomics in the Department of Pharmacy and Pharmaceutical Sciences and the Department of Oncology at St. Jude's Children's Research Hospital. He is also a member of the Hematological Malignancy Program at St. Jude Comprehensive Cancer Center. His research focuses on pharmacogenomics of pediatric cancer, especially acute lymphoblastic leukemia, and his group has led studies to identify genomic causes of a variety of drug resistance and toxicity phenotypes in children with ALL, many of which have been clinically implemented as described in his publications in JAMA, Nature Genetics, Lancet Oncology, and others. More recently, Dr. Yang has developed an imaging-based pharmacophenotyping platform to comprehensively define drug sensitivity profiles ex vivo for all leukemia patients at St. Jude, which are integrated with extensive genomic profiling to enable functional precision medicine. He is the recipient of the 2023 AACR Wong Ki Hong Award for Outstanding Achievement in Translational and Clinical Cancer Research. With that, I'd like to welcome Dr. Yang to discuss the pharmacogenetics of pediatric toxicity.
Thank you, Sarah, for your kind introduction and also for having me. Thanks very much for having me. I think this is my second or maybe third time giving this lecture. I did update some information from 2 years ago, so I hope that we will have a robust discussion in the end. What I want to do is to talk about pharmacogenomics in the context of a precision medicine, covering both discovery research as well as clinical implementation. So I'm going to start with some background on intrapatient variability in both drug-related traits as well as in genetics. And then the second part of my talk will focus on pharmacogenomics, its history since the 1950s, and talk about the differences and similarity between candidate gene-based versus a genome-wide discovery approaches. And I will go through a series of publications from others and ours, focusing on pharmacogenetics of therapeutic toxicity as a use case to illustrate the principle of pharmacogenetic research. And in the end, I'll hopefully have time to talk about the clinical implementation of pharmacogenetics and its impact for patients, either cancer or otherwise. So as a pharmacologist, we know there is a general positive correlation between drug dose on the x-axis and drug response on the y-axis. The more drug you give, usually you expect a stronger response. However, we also know that many patients do not follow this population average that deviates from this trend line. For example, the individual here showing at the bottom right. So even with a very high dose, the response is very, very low as indicated on the y-axis. So this will be considered as somebody resistant to this particular pharmacotherapy. But then you can also have individuals who are super sensitive to the drug to the degree such that they actually develop toxicity, such as individual here on the top left corner. So very low dose, a lot of response, but also a lot of side effects. So I think for the key of pharmacogenomics is really about the intrapatient variability in drug-related traits. We really want to identify the sources of this variability and try to use this knowledge to guide pharmacotherapy to achieve greater efficacy and less toxicity. So end of the day, it's about giving the right drug to the right patients. I think it's probably fair to say all human traits are at least somewhat related to genetics. This is a cartoon that was in the 90s, basically showing that there's nature versus nurture, both of which can affect drug-related traits. I mean, when we think about phenotypes related to pharmacotherapy, there are at least three. One's efficacy, whether or not you respond to the therapy in terms of your specific symptoms for which the drug was indicated, the side effects or toxicity, and also then the drug disposition and metabolism, which is the pharmacokinetics. So as I said, I think all three aspects of drug-related traits pull related to genetics. Now, what about the inter-patient variability in their genetic makeup? I think the lesson that we learned from sequencing human genome in the past 20-something years is that almost all human genes are polymorphic, meaning there's just incredible intra-individual variability in our DNA. I think the most common type of genetic variation is called SNP, single nucleotide polymorphism, and it's illustrated here. So we have sequence from three different individuals at this particular position. As you can see, they have three different bases, A, G, or T. So this single letter change in your DNA sequence is known as a single nucleotide polymorphism. So for every individual, we have somewhere between 4 to 5 million SNPs in our genome. I think 99.9% of the human genes have at least one SNP in the coding region. And about 50 to 60% of these coding polymorphisms are missense, meaning they actually change the amino acid sequence, therefore can potentially impact the function of the protein encoded by this gene. 99% of genes have at least one nonsense single nucleotide polymorphism. This is severe, right? Nonsense means that the protein cannot even be translated to its full length. So these are considered as very deleterious. So almost all genes in the human genome have some sort of variants with extreme deleterious effects. So I think this is quite a number of years ago, maybe 10 or 15 years ago, there was a huge exome sequencing project of thousands of individuals. So they're basically looking at all the coding sequence in the human genome and to identify the genetic diversity among different individuals. So it was reported at that time per person, every single one of us on this call will have something like 46 nonsense coding variants. So this is, as I said, this nonsense variants have a very severe functional consequences. And we also have about 52 frame shift or indel variants per person. Again, this causes a shift in your reading frame and therefore have very severe functional effects on the protein activity. So adding them together, I think per person, we carry about 100 deleterious variants in 100 genes, and they can have a very substantial germline defects. So this is just to underline the fact that there's a tremendous genetic variability across individuals. So the question for us as a pharmacologist is now what proportion of the variability in response to medication is genetic, or the drug-related traits, how much of that is explained by genetics? So every one of you probably familiar with Goodman-Gilman textbook in pharmacology. If you're not, maybe you should. This is the 11th edition. You had this really old study back in the 50s looking at pharmacokinetics of small molecule drugs in identical twins versus fraternal twins. As you can see, the PK parameters on the y-axis is almost identical across identical twins, but there's a significant variation was observed when you examine the same phenotype in fraternal twins. So this really points to the notion that the pharmacokinetic traits are highly heritable. Something like 75 to 85% of that is explained by genetics. What about pharmacodynamics? The way that cells respond to a medication. So back in maybe almost 20 years ago, Eileen Dolan's group from the University of Chicago did this really nice study to look at pharmacokinetics and its relationship to genetics. So what they did was to obtain cell lines from individuals from a large pedigree. As you can see, this is a 1, 2, 3, 4 generation pedigree. Every single individual in this family donated their peripheral blood to be banked for biological study. So what they did was to actually make a cell line using the B cells from each one of the individual in this family. And this particular study, they actually have mom, dad, and the kids, like a trio from this branch of the family. And they used they memorialized the B cells from these 3 individuals. And then they tested these cells for this sensitivity to daunorubicin, a commonly used anti-cancer drug. So what you can see is for the parents, their drug sensitivity is either very high, the red curve right here, or somewhat resistant, the blue curve on the top. And the child is in the middle, really reflecting that the genetics is inherited from both parents. So from similar studies with a larger number of individuals and samples, they estimate something like 30 to 70% of pharmacodynamic variability is driven by genetics. And this is validated by many other studies. But of course, I said the degree of to which genetics can explain this variability is different from drug to drug. So pharmacogenetics is aimed to connect genetic variation to drug response. We want to evaluate how an individual's genetic makeup corresponds to the response to a specific medication. And with the goal of pharmacogenetics is really to individualize medical treatments to the individual patients, and hopefully we will be able to maximize the effectiveness, but also reduce side effects of a specific medication. So when we look at pharmacogenetics and think about which genes or which proteins might actually affect drug-related traits, it's obvious that genes that encode proteins related to drug metabolism or disposition are obviously important. This includes all the drug metabolizing enzymes, drug transporters, but on the other hand, genes that encode protein that are targeted by your therapeutic agents are also going to be important, especially in the pharmacodynamic phenotypes. These are the receptors, enzymes that are inhibited by the drugs, cancer drug targets. Many of those fall in that category. So now I'm going to switch gear a little bit and then talk about pharmacogenetics, its history, different ways of doing pharmacogenetic study, and also walk through a specific example in the context of anti-cancer drug called valpyrin. So I think if we talk about the history of pharmacogenetics, probably can go back to the 50s. In 56, 56, 54, 57, there were 3 papers in JAMA and other journals describing that certain genetic traits or genetic conditions are linked to drug response. So, for example, individuals with the G6PD deficiency that was recognized back in the 50s, and these individuals actually are particularly susceptible to acute hemolytic anemia when they take anti-malaria drugs such as primaquine. So that's 56. That's 56, and 54 was discovered that some patients with low activity for the N-acetyltransferase (NAT), they're at a high risk for peripheral neuropathy if they're on isoniazid therapy. And there are other examples. The 57, for example, they look at relationship between cholinesterase and prolonged apnea with medications. So 57, based on these observations, it was argued that some idiosyncratic drug reactions were likely produced by specific genetic mechanisms. And this really provides the conceptual framework for pharmacogenomics. Like I said, it was published 1957. In 1959, the term pharmacogenetics was invented or coined. However, it should be noted that in the 50s, people obviously were not able to sequence DNA. We know it's genetics, probably based on familial studies, etc. But the actual genetic causes of this drug-related traits were not identified. So this is what I call the idiosyncratic area in the 50s. And moving on to the 60s and 70s, I think we kind of entered the biochemistry era of pharmacogenetics. So in the 50s, Price and Evans showed that isoniazid metabolism is actually a Mendelian trait by studying families who are exposed to this medication to look at their pharmacokinetic parameters. 77, it was showing that the metabolism of dibrisoquine is actually polymorphic, again using familial study design. And very similarly, in 1979, Michael Ekbom from Germany also showed the metabolism of spartine was polymorphic. But it was unclear as to the genetic causes of this variability, even though they suspect there's a genetic component. So in the 80s and 90s, with the advent of molecular genetics, molecular cloning, I think this field really exploded. In 84, the gene CYP2C19 was identified. Well, actually, the protein was identified. The gene was not cloned until 1993. In 1987 and 1989, a group in Switzerland really took the lead to start cloning a number of drug metabolizing enzymes, CYP2D6 in 87 and N82 in 89. Like I said, that really ushered us into an era of a lot of molecular genetic study focusing on drug metabolizing enzymes. So if 30 to 90% of the variability in drug response is genetic, how do we go about pinpointing the specific factors in our genome that drives the drug response and toxicity? I think there are two different approaches here. The first on the left is what we consider as a candidate gene approach. So for us to take a candidate gene approach, we have to assume that we actually know how a specific drug works, and we will only check the genes that are related to this drug. So for example, if we know which enzyme is responsible for the metabolism of a drug, we know which transporter is responsible for the disposition of the drug, then we can specifically focus on the metabolizing enzymes or drug transporters to understand if the genetic variation in those genes can affect the traits related to that specific drug. But a lot of times, I would say more often than not, we don't really know everything about a specific drug, how it works, how it's metabolized. In that kind of a situation, it's probably better for us to take a genome-wide approach, which is agnostic. So the idea here is we're going to look at the genetic variants across the entire genome without any assumption, and we just want to know in an unbiased fashion which genetic variation in which gene can be associated with drug-related traits. So what I'm going to do now is to use our experience in studying pharmacogenetics of thiopurine toxicity to illustrate the power of both candidate gene approach and also the genome-wide association study approach. So the disease that my lab is particularly interested is called acute lymphoblastic leukemia. This is the most common cancer in children, about 3,500 cases every year. In the US, it's a cancer that's for which chemotherapy was introduced back in the 50s. Methotrexate, mercaptopurine was actually one of the first cancers when treated with chemotherapeutic agents. So many of the drugs that we use to treat ALL are very, very old, going back to the 50s and 60s. And a particularly important class of drug for leukemia therapy is called thiopurines. And they are chemical analogs of the naturally occurring purine. And the structure for two of them are showing on the left. The molecule on the top is a 6-thiolguanine. The molecule at the bottom is 6-mercaptopurine. As I said, they're analogs of guanines. So both of them are incredibly important for curative ALL therapy in both children and adults. Patients are exposed to this drug on a daily basis for a year or year and a half, at least for children, and without which most of them will relapse. And myelosuppression is the main dose-limiting toxicity. It happens very, very common, and it's actually pretty challenging to manage patients who are that are on this drug. You have to titrate the dose very carefully based on WBC and ANC. If you dose reduce too much, you run the risk of exposing patients to less intense therapy, and which can lead to higher risk of relapse. But if you don't do a dose reduction enough, you expose them to excessive toxicity, which can cause treatment disruption of other chemotherapeutic agents, and that can lead to leukemia relapse as well. So it's a very fine balance between toxicity and efficacy. So for those of you who are not familiar with this drug, it's actually a prodrug. Mercaptopurine is showing here on the left. Once it's inside the cell, it actually goes through multiple enzymatic reactions to be converted into the active metabolites. So this is the base, and this base will need to be conjugated with ribose to form the nucleoside, and then phosphorylated monophosphates, diphosphate, and triphosphate. This triphosphate metabolite called TGTP is the active metabolite that gets incorporated into DNA double strand, which causes DNA damage and eventually cytotoxicity. This is the mechanism of action for thiopurine, at least in the context of leukemia. So back in the 80s, it was discovered that there is a human protein called TPMT, thiopurine methyltransferase. So this enzyme does one thing, and I think that's still the only function that we know about this enzyme. It adds a methyl group to mercaptopurine and produce methyl mercaptopurine, MEMP. And this is an inactive metabolite. So in a leukemia cell, you basically have this inactivation pathway going from top to bottom. And also you have this activation pathway going from left to right, mediated by HPRT and a bunch of other enzymes, which produce a TGTP, DNA damage, etc. So the balance between toxicity and the anti-leukemic efficacy is really the balance between those two pathways, right? Your inactivation pathway going from top to bottom, and your activation pathway going from left to right. So early I think it was the late 90s. Well, actually in the middle of the 90s. Mary Welling, Bill Evans at St. Jude, and Dick Weishaupt at Mayo Clinic started to sequence the TPMT gene, which encodes the thiopurine methyltransferase protein or enzyme. And they discovered there are 3 coding polymorphisms in the TPMT gene. This one, the G230AC, the 460, and 719. And all 3 coding variants lead to very severe loss of TPMT enzymatic activity, so there are loss of function variants. And this is clinically relevant because in the late 1990s, Mary Welling, together with Bill Evans and their collaborators here at St. Jude's, published a landmark study demonstrating the relationship between TPMT genetic variants and the risk of thiopurine toxicity. So on the y-axis is the cumulative incidence of severe myelosuppression from this drug. So these are leukemia patients on thiopurine therapy. So if you have 2 copies of the C allele, which causes loss of function, you basically have you have complete loss of TPMT enzymatic activity, and your inactive pathway is gone. So all you have is active metabolite, and this causes very severe toxicity, 100% risk of severe myelosuppression. If you're heterozygous, 1 copy of the normal allele, 1 copy of the defect allele, your risk of severe toxicity is only about 35%. If you're wild type, meaning you have 2 normal copy of the TPMT, your enzymatic function is normal, your risk of myelosuppression is only 7%. So this is this was tremendous discovery, because now you can use a TPMT genotype to predict who's going to have severe toxicity from thiopurine therapy before the drug is even given. So now with the pharmacogenetics, we have 2 different ways of giving thiopurine. The conventional way is to give everybody the same dose without considering their genotype, and let them crash and burn. Some of them will have severe toxicity because they have excessive exposure to the active metabolite. They have a lot more toxicity. But now with pharmacogenetics, you can genotype them up front, identify the wild-type individual, which is green, heterozygous, blue, and homozygous with the defective allele, which is red. So now you know they're gonna have different risk of toxicity, individualize their dose. If they're homozygous, you reduce their dose by 90%. If they're heterozygous, you reduce them by only 50%. And if they're wild type, give them normal dose. By doing so, you normalize their exposure to active metabolites. You essentially eliminate the excessive risk of toxicity for individuals carrying the risk allele, either heterozygous or homozygous, which is shown here at the bottom right. But one thing to point out is that even for individuals wild type TPMT genotype, 7% of them still experience severe toxicity. So that made us wonder there might be something else going on above and beyond what can be explained by TPMT. So Belinda is one of such patients. She was 5 when she was diagnosed with acute lymphoblastic leukemia in Malaysia. She is ethnic Chinese, and she went to Singapore for her leukemia therapy under the care of Alan Yo, who's a pediatric oncologist at National University of Singapore, also a long-term collaborator of mine. So for average Asian patients, showing here is their thiopurine dose across a couple of years for the LL therapy. You can see the dose titration. Usually you start low, but you gradually go up, and then it goes up and down because you have to titrate based on WBC and ANC. So usually for Asian patients, however, somewhere up between 50 milligram per meter square per day or 40 to 50 in that range. But for Belinda, it was actually very different. So immediately, within a couple of weeks, she started experiencing extreme toxicity from thiopurine. With a severe infection, her WBC was basically 0 for a couple of weeks, and she was in ICU. to the point that Alan Yu had to stop her thiopurine therapy completely for her count to recover. But once the count recovered, you have to give thiopurine again because it's such an essential drug. This whole toxicity and dose titration cycle just repeats itself. It actually had went on for months before Alan Yu finally determined that her tolerated dose was only about 2.6 milligram per meter square per day, which is much, much lower than average Asian kids, 50 milligram per meter square. So the first thing Alan thought about was maybe she was TPMT homozygous, loss of function variant, but turned out that she was wild type for TPMT. So clearly something else was going on. So for us to figure out this other genetic factors driving the severe toxicity, we decided to take a genome-wide association approach because outside of TPMT, we have no idea what other genes are important for this phenotype. The way we did this study was to collect two groups of individuals, patients who experience toxicity, patients without toxicity when they're on thiopurine therapy. And we got their germline DNA, extract the genetic material and the genotype using a high-density SNP array interrogating millions of single nucleotide polymorphisms per patient. And then we look at the genotype for its association with the phenotype of interest here, thiopurine toxicity, and we usually illustrate the results in the form of a Manhattan plot, which I'll talk about in a minute. So for this particular GWAS, we were able to get about 660 children with acute lymphoblastic leukemia. They were treated on thiopurine, and the clinical study was led by Doctors Mary Ralling and Smita Bhatia. And we also have a validation cohort, about 371 patients are treated on thiopurine here at St. Jude. So a phenotype of interest is 6MP dose intensity. So their prescribed dose is on the top, which is their tolerated dose based on WBC. And the protocol dose is at the bottom, so that's the standard dose. So if a patient has more toxicity, they cannot tolerate the standard dose, so you have to dose reduce based on WBC. So this percentage number will indicate the degree of toxicity. And we do this longitudinally for at least 6 months. And we genotype this individual for about 250,000 variants, mostly focusing on variants in the coding regions of the human genome. So this is the Manhattan plot summarizing the GWAS results. The y-axis is the log transform the p-value for the association between genotype and 6MP toxicity or dose intensity. Each dot here is a genetic polymorphism going from chromosome 1 to the sex chromosome. So you can the horizontal dashed line here represent the genome-wide significance threshold. Any dots above that line indicates a very strong, potentially reproducible genetic association. So the first finding here is TPMT on chromosome 6. So that's great because that's the positive control. We know TPMT already. But what's really cool was that we have a new dot on chromosome 13, and this signal is driven by a genetic variant in the gene called NUDT15. So here it shows the genotype, the association of NUDT15 genotype versus 6MP dose intensity. So for patients who are wild type for NUDT15 genotype, the average tolerated dose is about 85%, close to 90%. And if they're heterozygous for that particular variant in NUDT15, they can only tolerate about 65% of the standard dose, indicating pretty significant toxicity. And if they're homozygous for that variant, they can only tolerate about 8% of standard dose, requiring 92% dose reduction. So this is a very severe toxicity. And this is also validated. I apologize. This is actually, it's not TPMT. This is actually NUDT15. Sorry for the typo. And we validated this in the St. Jude cohort as well. So it turned Yeah, so this is the NUDT15 results. As I said, if they have a heterozygous or homozygous variant, they have a very severe toxicity from a therapy. And both in the discovery cohorts, about 660 patients, also validation cohort, about 400 patients with ALL here at St. Jude's. So this particular variant is actually in the coding region, is in exon 3 of the NUDT15 gene, and it causes a C2T change for this particular enzyme. So I don't want to go through the details because the paper is already published. We end up demonstrating that NUDT15 is a pyrophosphatase that is responsible for inactivation of the active metabolite TGTP. What it does is actually to convert the triphosphates active metabolites into the monophosphate metabolites, because only the triphosphorylated metabolite can get into DNA. This NUDT15 mediated reaction actually removes the active metabolite from the pool, and therefore can reduce cytotoxicity. So now if you have a genetic variant that causes a loss of function of NUDT15 enzyme, you basically destroy this inactivation pathway. You end up having a lot more active metabolite, TGTP. You have a lot more incorporation of this active metabolite in the double-strand DNA, more DNA damage, and more cytotoxicity. And this is consistent with the clinical observation of individuals with carrying these variants experiencing very severe toxicity from thiopurine. So what can we possibly achieve from a pharmacogenetic study? I think number one, pharmacogenetic study help us have a better understanding of the mechanism of specific medication, whether it's side effects or it's drug efficacy. I think this kind of knowledge can inform drug-drug interactions, can help us figure out better dosing and timing strategy. And I think the vast majority of pharmacogenetic studies will not lead to a clinically actionable discovery. But there is remote possibility that some of the pharmacogenetic discovery that we make might enable us to develop a clinical genetic test that can be used to guide prescribing. And I think the thiopurine example that I talked about is a good example of that, because of the effect sizes of the TPMT and also NUDT15 genetic variants are so huge on thiopurine toxicity. So they're actually clinically actionable. So on that note, I want to spend the last few minutes to talk about pharmacogenetics implementation in the clinic and how this actually changed the way that we administer pharmacotherapy. So actionable pharmacogenetic drug gene pairs have been known for quite a while. As I mentioned a few minutes ago, back in the 50s, we already know that G6PD deficiency is linked to hemolytic anemia, secondary to primaquine. And then, CYP2D6, TPMT, DPYD, HLA, they're all linked to various different medications. However, the use of pharmacogenetic information in clinical practice is actually quite rare and had remained rare for many, many years. And the question you want to ask is what's driving this sort of lack of action in the context of pharmacogenetics? So this study was published in Clinical Pharmacology and Therapeutics in 2017, characterizing the pharmacogenomic guided medication use with a clinical data repository. So they actually surveyed 132,000 individuals, covering more than 3 million hospital clinical visits. And there were something like 268,000 medication orders for 95 drugs, for which pharmacogenetic testing was already mentioned in the FDA-approved drug labels. And 49 of the 95 drugs were actually deemed as clinically actionable, which I'll talk about in a minute. So what the definition for that? So serving this huge clinical data repository, it was almost shocking to me and many others in the field of pharmacogenetics. Less than 1% of the prescription for these 95 drugs were actually accompanied by genetic testing. And 1.5%, even when the drug label made by FDA recommended or required genetic testing, it was just not down, right? Only 1%, 1.5% was actually accompanied by a genetic testing. So, like I said, what are the barriers to clinical implementation of pharmacogenetics? And this is another paper in many years ago doing a survey published in Clinical Pharmacology and Therapeutics, and specifically asking these questions. So 95% of the respondents thought that the process required to translate genetic information into clinical action was actually the most challenging piece when it comes to clinical implementation of pharmacogenetics. It was also mentioned that the genotype testing, although it's not necessarily very difficult to do, but the interpretation was very, very hard. It's hard to understand how the genotype information can be used to assign drug metabolism phenotype and how that will translate into what dose the patient will receive or what medication should be prescribed. And I think you also mentioned that the providing recommendation for selecting the drug gene pair to implement was a significant barrier because there are many drug gene pairs. So how do we prioritize? Do we do them all? Do we do specific selected pairs? So to address this challenge, Mary Redding and her colleagues in, I think was the end of 2009, start to think about a concept of developing clinical guidelines. In order to do that, they actually formed a consortium known as the Clinical Pharmacogenetics Implementation Consortium, or CPIC. The website is listed on the top right corner of the slides. So the goal of CPIC is to create, curate, and post clinical guidelines for gene drug practice. So basically, what it means is to give specific clinical recommendation for how to act on pharmacogenetics. So, for example, if a patient is homozygous for the NUDT15 star3 variant, what dose of thiopurine should that individual receive? So this is what CPIC wants to do, to link genetic information to clinical actions. So CPIC is a global organization with currently more than 780 members, including clinicians and scientists. They come from more than 590 institutions, 52 different countries. The guideline authors are only 23% of them are actually from outside of the US, including Europe, Asia, South America, Africa, Australia. So this is a truly international organization with a global impact in terms of, in the context of a clinical implementation of pharmacogenetics. So a very important thing that CPIC does is to assign evidence level for drug gene pairs. As I said, there are many drugs that can be metabolized by many enzymes encoded by different pharmacogenes. Not all of them warrant clinical action. So what CPIC does is to review the evidence very carefully using expert panel and assign levels, either A or B, which means that prescribing action is recommended for alternative therapy or individualized dosing. But for CPIC C and D, those are those are for drug gene pairs that are not ready yet for clinical implementation, whether because the effect size is too small or the evidence is not consistent across different studies. So this actually is extremely helpful because it provides a framework through which now we can start thinking about which drug gene pair or which drug gene association can be implemented in clinical practice. So the CPIC guidelines are posted on CPIC website. They're freely available. There are no limits on how you want to use them. They're peer-reviewed and published in Clinical Pharmacology and Therapeutics. It's a standardized format, which make it much easier for clinicians and scientists to understand across different drug gene pairs. As I mentioned already, they do provide grading for evidence and also recommendations. It can be updated on the websites as new evidence emerges. The development of this guideline follows very closely the National Academy of Medicine practice as published down below. So as of February 2025, there are 28 guidelines and 17 updates covering 34 genes and 160 drugs, some of which are listed here. As you can see, it really spans across many different therapeutic classes and areas. And the genes include the P450 enzymes, the drug transporters, and some drug targets as well. So it's estimated there are about 1,200 chemical entities that are approved as drugs worldwide. And we have about 18,000 genes. So if you draw a Venn diagram, there are the one in the middle. So 31 genes for 87 drugs with a clinically actionable drug gene association. And this translates to about 18% of all prescriptions in the United States. So this is actually a very significant proportion of individuals who might benefit from pharmacogenetics. So I'm going to skip this slide. This is really just to show you where you can find this information on PharmGKB, which drugs have a different pharmacogenetic label. And to do this at St. Jude, we have developed a clinical trial called PG for Kids. The goal is to migrate pharmacogenetic testing results from a laboratory into a research laboratory into routine patient care and to be available preemptively. This is a process for PG for Kids. Patient consents, get their blood sample. We perform genotyping for about 1,100 genes, and they go into a research database. And then based on the evidence review, we decide which drug gene pair should be clinically implemented. And then we, only for those, we return results to the prescriber and also patients. And we have developed a clinical decision support system to enable implementation in the electronic health records, which results will be available at the time of prescription. So we have implemented 17 genes linked to 275 medications to individualize pharmacotherapy here at St. Jude, primarily in accordance with the CPIC guidelines. These are the genes that we implemented so far. From 2011 all the way to 2025. So as of just a few months ago, we have enrolled over 7,000 kids in the PG for Kids clinical study, and 95% of them have actionable pharmacogenetic finding. You can see that it really depends on the number of pharmacogenetic variants they have. But like I said, the vast majority of them have something that can be clinically acted upon. So this is the CDS, clinical decision support, that we develop to enable implementation in the EHR environments. This is an example of that. This is a pre-test alerts, really contain prescribing and testing recommendation if a patient has not been genotyped yet. So for example, if somebody wants to prescribe a medication that's metabolized by CYP2D6, a warning sign will pop up to say, well, you need to prescribe, order a genetic test for 2D6 before you prescribe that drug. And this is a warning after the test is performed, really to say that, oh, the testing results indicate that this patient is a poor metabolizer. If you're prescribing codeine, then you have to consider individualized dosing based on the pharmacogenetic results. So just to summarize, I think I hope I've convinced you that pharmacogenomics is a very important part of precision medicine. It is a branch of genomic medicine with the aim to identify genetic basis of interpatient variation in drug response and toxicity. Most of the candidate gene studies focus on P450 enzymes, drug transporters, and they are obviously involved in drug disposition and metabolism. And genome-wide association studies are particularly powerful because they can identify genetic variants affecting drug response or toxicity in an agnostic fashion and comprehensively. And CPIC, the Clinical Pharmacogenetic Implementation Consortium, is this organization that's responsible or driving the development of clinical guidelines to use pharmacogenetics clinically. I think many barriers still exist in pharmacogenetics-driven precision medicine, especially in the implementation space. But I'm very hopeful that with better education and better discovery science, pharmacogenetics will become routine in pharmacotherapy in the near future. As a matter of fact, it has been routine practice here at St. Jude already. So I need to thank a number of individuals who contributed to the therapeutic story that I talked about. Mary Welling, Bill Evans, Katie Cottle, Serene Haidar, James Hoffman, who have been instrumental in developing clinical guidelines for pharmacogenetic implementation. Our collaborators here at St. Jude and also globally. I also need to thank my funding sources. So I will just one photo of Linda. She turned out to be homozygous for the NUDT15 variant. It's actually quite nice for a laboratory scientist like me to know that our discovery actually made a huge difference for patients like Belinda. So I'll stop here and be happy to take a few questions.
All right. Thank you, Jun. Very interesting story, and I also enjoy it when we can see where our research is impacting clinically and helping people directly. I think it's as clinical pharmacologists, one thing we're really looking at is that outcome result. So yeah, so we have a couple of questions in the Q&A box. I encourage others to submit questions there. The first, we know that genetics have a strong predictive power on how patients respond to drugs. However, as you mentioned, the implementation in the clinics is very rare, especially in developing countries and more rural areas, I would say even in the US. What can we as clinical pharmacology researchers do to increase the acceptance of pharmacogenetic testing in the clinical space?
Yeah, that's a really good point. I think that's a question that we get asked a lot. I think there are a few things that we can do. Well, of course, depends on the role of that individual. So I think we definitely want to have, as a clinical pharmacologist, we need to do more discovery work to really better understand the genetic basis of interpatient variability in drug response or toxicity, right? Without the discovery research, there's nothing for us to implement. But in the meantime, when a drug gene pair is already, the association is well established, why hasn't been implemented? I think there are a number of different reasons. One is education. I think a lot of time for your general practitioners, they are very, very busy, and they don't necessarily very well versed in the genetic literature. They might have heard about pharmacogenetics, but they're not exactly sure how that impacts their clinical care. So I think the education needs to start early, maybe even during medical school. They need to understand that pharmacogenetics has real patient impact. And once they have that awareness, I think they will become a lot more receptive to implementing pharmacogenetics. Another important aspect about this is I think it's getting easier to order a genetic test. But once the genetic testing results are back, it's actually not very easy to understand and to know what to do. especially in the community setting, whether it's rural or inner city. The general practitioners, like I said, they don't read pharmacogenetic literature all the time. If somebody is heterozygous or homozygous for a gene means not very much to them. So without a carefully developed guideline, it's very hard for them to actually use that in clinical practice. But I think what has changed in the past few years are like phone apps or technology developments that will make it much easier for them to just to say, okay, I have this phone app now, whether it's in their EHR or whatnot, and the genetic testing results come back, and then they will provide a very specific recommendation on what dose should be used. So that becomes much more convenient for the practitioner to understand and also to implement. And here at St. Jude, we also have pharmacists who facilitate that process. So I think pharmacists actually can play a very important role in clinical implementation of pharmacogenetics because in a way, they're ones who are dispensing the drug. They're the authoritative voice in that space.
Thanks. I think that leads into, some of the other questions here. Specifically, you mentioned that, incorporating it in earlier into medical education. So how is pharmacogenomics currently being included in medical education? And this attendee points out that medical doctors seem to be more resistant than pharmacists to implementing pharmacogenomics.
Yeah, I would say that the education on pharmacogenomics is definitely not very systemic. Right? I mean, it's just some medical schools, depending on who's the dean or whatnot, they might be more proactive about pharmacogenetics or genetics in general. I think my impression is that the pharmacists think a lot more about drugs, and the medical doctors, they think a lot more about the diseases. So they are sometimes very receptive to implementing genetics in the context of a disease diagnosis. So if it's a rare genetic disease, they don't necessarily have a lot of time and a lot of problem implementing it because it will help them to arrive at diagnosis. But if it's a medication, especially as a medication that's been used for a long time, they oftentimes feel like, oh, I can do this. I can just dose titrate, and it should be okay. But the example that I showed for thiopurine, yes, you can dose titrate, but it took 3 months for them to arrive the stable dose, and during which time the patient was at risk of very severe myelosuppression, and she could have had very bad consequences, right? I mean, had she not been under the care of a very careful physician. So I think in 2026, it's hard for us to argue that, oh, we know pharmacogenetics, but we'll still let them experience the toxicity and then just come back and dose titrates. I think that sort of argument is not very compelling anymore in 2026. That's my argument.
Yeah, and then there's a couple of questions or comments looking at really, advocating and how can we advocate for our patients and to expedite pharmacogenomic implementation. And then also, looking at, presenting CPIC presentations at medical conferences and to patient-focused webinars, more hitting the community. Are there plans or activities in the works that are aiming to do that?
Yeah, so CPIC is actually very proactive about it. CPIC organizes the conferences together with the PGRN, Pharmacogenetic Research Network. Then they offer CME credits. So they actually develop specific educational tools to talk about pharmacogenetics implementation. And they also have free videos online they put on YouTube for people to watch if they're interested, talking about specific drug gene pairs. So I think that information is out there. I think, but you're absolutely correct. I think we can do more to educate our practitioners, pharmacists, and patients as well.
We're just up at 1 o'clock here, Eastern time. I just want to ask one more question that popped up in the chat about how the module you discussed alerting practitioners about the need of pharmacogenetics data before prescribing is working, and whether these tests are being reimbursed by insurance.
Yeah, so St. Jude has a very different financial model, so we don't necessarily we cover a lot of the care expenses if it's not covered by insurance. But I think a lot of times, but in other many other places, this is the local coverage determination. So I think pharmacogenetic testing is reimbursed, although the degree of reimbursement and the pattern differs by the location. But the local coverage determination is a mechanism for pharmacogenetic reimbursement. And we implement the clinical decision support system in our APIC context and works pretty well. I think the acceptance rate, or in terms of how many times they actually act on it, differ by physician, is generally pretty high here at St. Jude, just because they heard about pharmacogenetics all the time, every day, multiple times a day. But in other places, I think it might be a little bit different.
Thank you very much for an excellent talk. I'm sure we have a lot to move forward here, and hopefully that everyone on the call now will be advocating for the use of pharmacogenomic testing in their patients. So thank you, Dr. Yang.
Thanks again for having me.
With that, I'll close today's webinar. We do not have a webinar scheduled for next week. Our next CPTN webinar will be on Wednesday, February 4th, featuring Dr. Diane Mold, talking with a talk entitled Combining Bedside and Clinical Research Data to Inform Disease Progression and Outcomes. So we look forward to seeing everyone then. Thank you.